Welcome to G Fun Facts Online. Today, we're diving deep into the code of life, into a mystery that has puzzled scientists for decades. Imagine you have the complete blueprint for a human being, the entire genome. You'd expect every part of it to be crucial, right? Well, for a long time, we thought only 2% of our DNA actually did anything important, the part that contains the recipes for proteins, the building blocks of our bodies. The other 98%? It was dismissed with a rather unflattering name, junk DNA. This vast, enigmatic territory was considered an evolutionary junkyard, filled with useless leftovers. But what if that junk wasn't junk at all? What if it was the most important part of the manual, a hidden layer of control that scientists are now calling the dark matter of the genome? This isn't just a simple correction in a textbook. It's a revolution that is changing everything we know about our health, our evolution, and what it means to be human. The story of how junk DNA became the hottest field in genetics begins in the mid-20th century. Scientists like J.B.S. Haldane were wrestling with a problem. If every single letter of our DNA was essential, the number of harmful mutations we'd accumulate would be devastating. This led to the idea that most of our genome had to be non-essential. The term junk DNA was officially coined in the 1960s, but it was geneticist Susumu Ono who truly popularized it in 1972. Ono pointed to something called the C-value paradox. This is the bizarre fact that the size of an organism's genome has no relationship to its complexity. For example, some salamanders, and even onions, have genomes many times larger than ours. Are they more complex? Unlikely. Ono argued this extra DNA must be non-functional baggage. He saw our genome as an archaeological dig site, littered with the fossilized remains of extinct genes, duplicated and then silenced over millions of years. Then came the discovery of introns in the 1970s. These are long, non-coding stretches of DNA that interrupt our genes and get snipped out before a protein is made. It seemed like more evidence for the junk theory. To top it all off, scientists discovered transposable elements, or jumping genes. These are DNA sequences that can copy themselves and hop around the genome. By the 1980s, it was clear that nearly half of our DNA is made of these elements. Famous scientists like Francis Crick even called them selfish DNA, genomic parasites that exist only to replicate themselves, with no benefit to us, their host. But even then, some scientists were skeptical. Could evolution really be that wasteful, carrying around so much useless code for millions of years? They suspected this dark matter might have subtle, hidden functions, and they were about to be proven right in a spectacular way. The game changer was the ENCODE project, which stands for the Encyclopedia of DNA Elements. Launched after the Human Genome Project was completed in 2003, its goal was audacious: to create a complete parts list of every functional element in our DNA. In 2012, after nearly a decade of work by hundreds of scientists worldwide, the results were published. The headline was a bombshell. A staggering 80% of the human genome has some kind of biochemical function. The media declared the death of junk DNA. The era of the dark genome had begun. ENCODE identified millions of previously unknown gene switches, regions of DNA that control when and where our genes are turned on and off. This was huge, because we already knew that about 90% of the genetic variations linked to common diseases were located outside of protein-coding genes. Suddenly, ENCODE provided a map to understand why those disease-linked variations were likely breaking these newly discovered switches. However, the 80% functional claim sparked a fierce debate. Critics argued that ENCODE used a very broad definition of function. They classified any piece of DNA as functional if it showed biochemical activity, like a protein binding to it. But does that mean it's truly important for the organism? The critics argued for a stricter, evolutionary definition. For a sequence to be truly functional, it must be conserved by natural selection, meaning that changes to it are harmful and get weeded out over time. By that standard, only about 5 to 10% of our genome is conserved. Proponents of ENCODE countered that their goal was biomedical discovery, not evolutionary theory. If a region is active, it might be relevant to health, even if it's not perfectly conserved. Despite the controversy, ENCODE fundamentally shifted the conversation. It revealed that the genome isn't a simple string of genes littered with junk. It's a complex, multi-layered information processing system. So what does this genetic dark matter actually do? Let's think of our genes as the instruments in an orchestra. The protein-coding genes are the violins, the cellos, the trumpets, but who tells them when to play, how loudly, and in what combination? That's the job of the non-coding DNA, the conductor of the genetic orchestra. Much of this non-coding DNA consists of regulatory elements. These are short DNA sequences that act as docking sites for proteins. The most common types are enhancers, which act like accelerators, dramatically boosting a gene's activity. Then there are silencers, which are the brakes, slowing or stopping a gene. And finally, insulators, which act as barriers, organizing the genome into neighborhoods and making sure an enhancer for one gene doesn't accidentally turn on its neighbor. The interplay between these elements is what allows a single genome to create hundreds of different cell types, from a neuron to a skin cell. It's not just the linear sequence that matters. We now know the genome is folded into an intricate 3D structure inside the cell nucleus. DNA is organized into loops, which can bring an enhancer that's thousands of letters away, right next to the gene it needs to activate. These folds create insulated domains, ensuring that regulatory signals don't cross wires. A mutation that disrupts this 3D folding can be just as catastrophic as a mutation in a gene itself. Perhaps one of the most stunning revelations has been the RNA revolution. We used to think of RNA as just a humble messenger, carrying instructions from DNA to the protein-making machinery. We now know that's only a tiny part of the story. A huge portion of our so-called junk DNA is transcribed into a vast and diverse world of non-coding RNAs, molecules that are not translated into protein but have critical jobs of their own. There are long non-coding RNAs, or LNCRNAs. These are the Swiss Army knives of the genome. Some act as signals, telling the cell how to respond to a stimulus. Others act as decoys, like molecular sponges that soak up other proteins to stop them from doing their job. Some act as guides, leading enzymes to specific spots on the genome to turn genes on or off. A classic example is a LNCRNA called EXIST, which blankets one of the two X chromosomes in female mammals, completely silencing it to ensure a proper dose of genes. Then there are microRNAs, or miRNAs. These are tiny RNA molecules, just 22 nucleotides long, but they are giants of gene regulation. They bind to messenger RNAs and either mark them for destruction or block them from being made into proteins. It's estimated that miRNAs regulate at least half of all human genes, involved in practically every biological process. This hidden world of RNA adds an entirely new layer of complexity to how our cells operate. The dark matter of the genome isn't just a regulator, it's also a powerful engine of evolution. The very things that made it seem like junk, its repetitive nature and the selfish jumping genes, are actually sources of incredible genetic innovation. Remember those transposable elements, or jumping genes, that make up nearly half our DNA? Our cells are in a constant arms race with them. These elements want to jump and spread, which can cause harmful mutations. In response, our genome has evolved sophisticated protein armies to find and silence them. This battle has been a major driving force in the evolution of our gene regulation systems. But sometimes, a parasite can become a partner. Over millions of years, our genome has learned to tame these jumping genes, co-opting them for new functions. This is a process called exaptation. A transposable element might land near a gene and, because it contains regulatory sequences, it can suddenly give that gene a new on-off switch. For instance, a class of TEs called MER41B has been repurposed to create new regulatory elements that are crucial for our innate immune response. Some have even been fully domesticated into new, essential genes. The most famous example is the RAG1 gene, which is vital for our adaptive immune system, the part that creates antibodies. And where did it come from? It's derived from an ancient jumping gene. It's as if we took a piece of a computer virus and turned it into our main antivirus software. And it gets even stranger. About 8% of our genome is made of HERVs, or human endogenous retroviruses. These are the fossilized remains of ancient viruses that infected our primate ancestors millions of years ago and stitched themselves into our DNA. Most are broken and inactive, but some have been repurposed in truly mind-boggling ways. The most incredible example is a set of genes called syncytins. These genes are absolutely essential for the formation of the placenta, the organ that connects a mother to a developing fetus. And they are derived from the envelope genes of ancient retroviruses. The very property that allows a virus to fuse with a cell was repurposed to help placental cells fuse together. It appears this happened independently in different mammals, a stunning case of evolution finding the same brilliant solution multiple times by recycling ancient viral junk. So this dark matter isn't a graveyard. It's a dynamic, creative playground where evolution experiments and forges new traits. If this dark matter is so important for regulation and evolution, it stands to reason that when it goes wrong, it can cause disease. And that is exactly what we're finding. Over 90% of the genetic variants linked to common diseases from large-scale studies are found in non-coding DNA. These are broken switches, faulty wiring in our genetic control panel. A mutation can create or destroy a binding site for a regulatory protein, causing a gene to be turned on when it should be off, or vice versa. This new understanding is shedding light on countless diseases. In cancer, for example, many tumors are driven by mutations in the promoter of a gene called TERT. This non-coding mutation keeps the gene hyperactive, allowing cancer cells to become immortal. In neuroscience, defects in non-coding DNA are being linked to autism, schizophrenia, and even language disorders because brain development relies on exquisitely precise gene regulation. The same is true for cardiovascular and autoimmune diseases, where non-coding RNAs have been found to be key players in disrupting the normal function of the heart and the immune system. The incredible part is that this new knowledge isn't just academic, it's opening up a new frontier of medicine. For the first time, we can think about targeting the root causes of disease by fixing the faulty regulation. Many disease-causing proteins are considered undruggable by traditional drugs, but we can now target them indirectly by going after their non-coding RNA regulators. One approach uses something called antisense oligonucleotides, or ASOs. These are short, custom-designed molecules that bind to a specific rogue RNA and mark it for destruction. Several ASO drugs are already on the market. Another strategy uses siRNAs, or small interfering RNAs, which can silence a specific gene with high precision. And, of course, there's the revolutionary gene editing tool CRISPR, which holds the promise of directly editing and correcting mutations in these non-coding switches right in our DNA. Beyond treatment, non-coding RNAs are also proving to be powerful biomarkers. They can be found in our blood and urine, which means they can be detected with a simple liquid biopsy. Their levels can signal the presence of a disease long before symptoms appear, predict how a disease will progress, and monitor how a patient is responding to treatment. This is the future of personalized medicine. We have come a long way from dismissing 98% of our genome as junk. We've journeyed from a static blueprint to a dynamic four-dimensional operating system. The dark matter of the genome, once an enigma, is now seen as a bustling command center, an engine of evolution, a source of disease, and a powerful new target for medicine. And yet, we have only just begun to explore this vast, uncharted territory. Answering the remaining questions: How do millions of switches coordinate the symphony of life, and how can we safely and effectively manipulate them? Will be the great challenge for the next generation of scientists. The story of junk DNA is a powerful reminder that in science, today's mystery is often tomorrow's revolution. The secrets hidden in this genetic dark matter are no longer just a curiosity. They hold the key to a future where we can better understand, treat, and perhaps even prevent our most devastating diseases. The age of the non-coding genome has truly begun,